Hello everyone, welcome to Brain Blitz Audios. In today's episode of NEET preparation, we'll be dealing with previous year questions of biology in medical entrance examinations in India. Today, we're looking at the chapter Photosynthesis in Higher Plants. Now this chapter is simple and short, but the concepts in it are very important. Therefore, there are many questions in here that are often asked. This chapter is the 13th chapter in the grade 11 syllabus for CBSE. I urge you to watch the full video so that you do not miss any of the questions given here. Having said that, let's move on to our first question. In C4 plants, the first carbon dioxide acceptor is A. Pyruvate, B. Phosphoenol pyruvate, C. Ribulose bisphosphate, D. Ribulose 5-phosphate. So the question is asking us to find out the first carbon dioxide acceptor in C4 plants. So when plants do photosynthesis, the CO2 from the atmosphere is accepted by a compound. It differs in C3 and C4 plants. Now C4 plants, these are adapted towards dry tropical regions. So therefore they have a special mechanism for photosynthesis. Examples of C4 plants include sugarcane, maize, corn, etc. Now, the speciality in C4 plants is that they undergo double fixation of carbon dioxide. Now, this double fixation happens in two parts of the leaf, the mesophyll cells and the bundle sheath cells. Now, the mesophyll cells fix carbon dioxide without the use of the enzyme rubisco because it's not present there. And rubisco is the prime reason behind photorespiration in plants. So basically what they're doing is they're fixing the carbon dioxide such that in the mesophyll cells, which do not have rubisco, and then under secure environment in the bundle sheath, they again fix it and then start producing food out of it. So there is no photorespiration in C4 plants, and this is more efficient. So if rubisco isn't the enzyme present, then what enzyme and what acceptor is present? So CO2 is converted by the acceptor into carbonate. It's CO3 minus. Now, this hydrogen carbonate, along with the carbon dioxide acceptor, undergoes this reaction. So it's CO3 minus plus our acceptor under the presence of PEP carboxylase gives you oxaloacetic acid. This oxaloacetic acid is converted into malic acid, and the malic acid enters the bundle sheath cells and are broken into CO2 and other 3-carbon compound. And that CO2 enters the C3 cycle inside the bundle sheath cells and it starts producing food. Now, PEP carboxylase is an enzyme. And so when the term carboxylase is present, the term refers that the hydrogen carbonate ion, which is present in the acceptor, that is the prefix, will be fixed. So here it is PEP that is the first carbon dioxide acceptor. And this PEP can be written in its full form as phosphoenol pyruvate. So option B is the right option. Another way to think of this is that the first carbon dioxide acceptor will be present in the mesophyll cells. So we just need to find out which of these is present in the mesophyll, and that will give you the first carbon dioxide acceptor. Options A, C, and D are incorrect because pyruvate, ribulose bisphosphate, and ribulose 5-phosphate are all present in the C3 cycle. And this C3 cycle happens in the bundle sheath cells. And so one of the fixations happen there, but there's another carbon dioxide fixation which happens in the mesophyll. So therefore, the only enzyme here that is the first carbon dioxide acceptor is phosphoenol pyruvate. And after analyzing the processes inside the mesophyll, we find out that phosphoenol pyruvate, which is referred to as PEP, is the right option. So in C4 plants, phosphoenol pyruvate is the first carbon dioxide acceptor, and that is option B. Let's move on to the next question. In photosynthesis, splitting of water and release of oxygen occurs during photolysis, red drop, Pasteur effect, Calvin cycle. So photosynthesis itself has two reactions, generally, the light reaction and the dark reaction. So the light reaction enables the formation of ATP and NADPH. So it does that in the presence of a photosystem titled photosystem 2. So in order to convert NADP plus, it needs an electron, and in order to get this electron, what it does is it initiates photolysis of water, and the reaction is 2H2O gives 4H plus O2 plus 4 electrons. So as you can see, the electrons are used to satisfy NADP plus, and one of the hydrogens is taken there in order to give NADPH, in order to form NADPH, and the oxygen passes out through the stomata, so oxygen is released and water is split. So the right answer is option A, photolysis. Let's move on to the next question. Blackman's law of limiting factor is applied to A, respiration, B, transpiration, C, photorespiration, D, photosynthesis. Now, what is the law of limiting factors? So the law of limiting factors states that if a chemical process is controlled by many factors, which here means more than one, then the rate of that chemical reaction will be determined by the factor among the many factors present in limited quantity. So this is the statement of the Blackman's law of limiting factors. So suppose we have photosynthesis. It depends on the sunlight, water, nutrients, CO2, etc. And suppose the plant lives in an area where there's a scarcity of water. So the rate of photosynthesis will always depend on the amount of water. So therefore, even if all the other factors are provided excessively, the rate of photosynthesis will still depend on the quantity of water present. So that relation, which is found in photosynthesis, makes photosynthesis an application of Blackman's law of limiting factors. So therefore, option D is the right option. Option A is respiration. Now, respiration in living organisms is usually found in controlled environments, such as inside the body and the cells which already have the oxygen and the nutrients all supplied by the blood. So option A is incorrect because this happens in a controlled environment. Option B, transpiration, is incorrect because transpiration is a physical process. You know, when water evaporates through the leaves and it's sent out through the stomata, it creates a transpirational pool, which is a physical process by which water is sucked in from the lower half of the tree. So transpiration is physical. And option C, photorespiration, it happens in the presence of light and O2. And since photorespiration always happens when O2 is around, there's only one factor, that is light, which may vary consistently as the reaction takes place. Because if O2 were to fluctuate, then photorespiration would stop when there's less O2. So option C is incorrect. The right answer is option D, photosynthesis. It is dependent on a variety of factors, and if one of them has a lesser quantity, then the rate of photosynthesis will be affected by it. It has been found experimentally. Let's look at this question. Hill reaction occurs in high altitude plants, total darkness, absence of water, presence of ferricyanide. So, what is the Hill reaction? The Hill reaction in photosynthesis is the same thing as the light reaction, the half of the photosynthesis which is dependent on light, where photolysis occurs. So over here, the most important parts are light and water, and then an electron acceptor. And for photosynthesis, that is found in PS2. The whole reaction is taking place in PS2, photosystem 2. So one of these options has all of the conditions and therefore is the right answer. Let's find out which one. Option D, presence of ferricyanide. Now, ferricyanide is an electron acceptor, but here's the catch. It's present in photosystem 1, while this reaction happens in photosystem 2. So option D is incorrect. Option C, absence of water. It means that there is no water. So without water, there is no photolysis. Without photolysis, there is no replenishment of electrons. There is no release of oxygen. There is no light reaction. So option C is incorrect. Option B, total darkness. This is the opposite of the condition light. So total darkness is without light. The area does not have any light, which is detrimental because light reaction is dependent on energy received, the light that they receive. So option B is incorrect. The right answer is option A, high altitude plants. High altitude plants have plenty of light. They also receive water in the form of snow or rain, and these plants and plants of all kinds must have chlorophyll, which have PS2 in them. So option A is the right option. Let's look at this question. Which one of the following categories of organisms do not evolve oxygen during photosynthesis? So we've always heard of plants evolving oxygen during photosynthesis, but there are some species, some organisms, which do not evolve oxygen. We need to find out which one. Now, oxygen is evolved by the splitting of water. So that means in photosynthesis that happens in the presence of chlorophyll, the source of the electrons is water. So if we can find out which of these organisms do not have chlorophyll in them, then we will be able to say that they do not evolve oxygen during photosynthesis. Let's look at our options. Option A, red algae. This is named due to the color of the algae, which is red. That is due to other pigments, but red algae also has a type of chlorophyll known as chlorophyll D. So therefore, it is bound to form oxygen because chlorophyll has its source of electrons as water. So option A is incorrect because they do evolve oxygen. What about option D, blue-green algae, also known as cyanobacteria? these are a special class of bacteria which uses chlorophyll A along with other pigments in order to prepare food for themselves. So that means chlorophyll A also has its source of electrons as water, so it must evolve oxygen. So option D is also incorrect. What about option C, C4 plants with Kranz anatomy? Now in C4 plants, the only difference is the anatomy of the leaf, the positioning of the enzymes such as peptase and ribisco, and the non-evolvement of oxygen so as to avoid photorespiration. However, both C4 and C3 plants belong to angiosperms, and they use the pigment chlorophyll B extensively. So therefore, option C is also incorrect. The right answer is option B, photosynthetic bacteria. Now, these are very different from cyanobacteria. Cyanobacteria is a separate class of their own. These are other kinds of photosynthetic bacteria, and they use the pigment bacteriophyophytin. And this pigment uses H2S as its source of electrons and not water. So therefore, they will not evolve oxygen. They will evolve other compounds such as S2. So that's why option B, photosynthetic bacteria, is the right option, because it does not use chlorophyll. It uses bacteriophyophytin, which is also known as bacteriochlorophyll. And this pigment requires a different source of electrons than water. So there will be no evolvement of oxygen as a result of photolysis of H2S. So therefore, option B is the right option. Now, let's look at this question. What is PAR range? And we're given ranges from 200 nanometers to 800 nanometers, 400 nanometers to 700 nanometers, 350 nanometers to 550 nanometers, 600 nanometers to 100 nanometers. So what is PAR? PAR stands for photosynthetically active radiation. So this PAR refers to the amount and the wavelength of radiation in which the plants get enough energy in order to do photosynthesis. So we need to find out the range of the wavelength in which this occurs. Now, in order to make our work easier, I've embedded a graph which shows you the absorption spectra of various kinds of chlorophyll and other pigments. So as you can see, chlorophyll A has the spike at both ends. Chlorophyll B also has a greater spike at both ends, and carotenoids occupy a small spike in the middle. So as you can see, the lowest of these, the lowest wavelength in which all these pigments accept energy from the sun is at 400 nanometers, and they end at 700 nanometers. So therefore, we can say that option B, 400 nanometers to 700 nanometers, is the right option. Of course, you can also deduce it from the term photosynthetically. So it is present in photosynthesis, and photosynthesis means synthesis of food from light, and the wavelength of light is 400 to 700 nanometers. But by showing this, we have given conclusive proof in order to find out the correct range for photosynthetically active radiation. So option B is the right option. Now, this question states, through the use of oxygen 18, which is heavy oxygen, scientists have found that the oxygen released under photosynthesis comes from the molecules of A, carbon dioxide, B, water, C, glucose, D, chlorophyll. So we need to find out from where does oxygen evolve during the process of photosynthesis. Now, through the oxygen 18 or heavy oxygen is actually not needed in this question, but that way you can understand how scientists find out certain elements in processes. They use the heavier isotope of the element and then they find it out. And coming back to our question, we need to find out the oxygen is released from which of the molecules. So if you refer our second question, it said that photosynthesis contains two reactions, the light reaction and the dark reaction. And in the light reaction, NADP plus is converted into NADPH. So we're forming energy currencies. And in order to make up the loss of electrons and stuff, in order to put in more electrons, they do a process known as photolysis. And this photolysis is basically the splitting of water in order to get hydrogen ions, oxygen gas, which is released, and then 4 electrons. So they use the electrons of the hydrogen plus, and then they remove the oxygen. So that oxygen is released, and that is the oxygen that we breathe. So therefore, option B, water, is the right option. Carbon dioxide in photosynthesis becomes the food or carbohydrates that plants form. So option A is incorrect. Option C, glucose, is the end product which is stored in plants in the form of starch. So therefore, option C is incorrect because this is already formed. They wouldn't break it further in order to release oxygen because this is the final product that plants need. And option D, chlorophyll, acts as a catalyst. So for photolysis and all the other reactions, the reduction of carbon dioxide, etc., that happens inside the leaf, chlorophyll is just a catalyst, and it is the medium by which sunlight is absorbed and then transferred from light energy into chemical energy. So therefore, option D is also incorrect. The right answer is option B, water, because the oxygen released happens to come from photolysis where water is split. So it comes from the molecules of water. Now, in this question, we're asked to find out the incorrect statement. So, big note, we need to find the incorrect statement and not the correct one. So over here, the statement which is wrong, we should mark that as the right answer. So let's look at our first option. C4 pathway for CO2 fixation were discovered by Hatch and Slack. This statement is true because another name for the C4 pathway is the Hatch and Slack pathway. And it was given that name due to the fact that Hatch and Slack discovered the C4 pathway in the plants, sugarcane, maize, and other plants of the same anatomy. So option A is incorrect because the statement A is true. Option B, CO2 is essential for photosynthesis. Now, in photosynthesis, CO2 is reduced to form carbohydrates. So therefore, it is an essential aspect of photosynthesis. Therefore, this statement is true. So therefore, option B is incorrect. Option D, phloem is the principal pathway for translocation of solutes. Translocation of solutes refers to the transport of the food prepared in the leaves. And food prepared in the leaves passes through the connective tissue, the vascular tissue known as phloem. The xylem is used for the transport of water, and phloem is used for the transport of food. So therefore, option D is also incorrect because this statement is also true. That makes option C, addition of sodium carbonate in water retards photosynthetic rate in Vallisneria, as the right option because that statement is incorrect. And the reason why it is incorrect, there are two things. One thing is that sodium carbonate acts as a source of CO2. And number two, the addition of sodium carbonate in the plant Vallisneria actually accelerates the photosynthetic rate instead of retarding it. So when you add sodium carbonate, instead of retarding photosynthetic rate, you get the exact opposite effect. So therefore, the statement given is false, and since we're asked to find the incorrect statement, we know that this option, option C, is correct for this question. Now, this is the next question. The family in which many plants are C4 type: A. malvaceae, B. solanaceae, C. cruciferaceae, D. graminae. So we need to find out which of these families have plants which contain C4. Now, the important thing to note here is that C4 pathway is found in about 1,500 species, which belong to 19 families, all of which are under angiosperms. However, the crazy thing about C4 type is that most of them are present in monocots, such as sugarcane, maize, etc., which are present mostly in the family known as Graminae. So therefore, option D is the right option. Option A, Malvaceae, contains plants such as okra, which are C3. So most of it is made up of C3. So option A is incorrect. Option B, Solanaceae, contains plants such as the potato, and most of Solanaceae also are dicots, which follow the C3 pathway. So option B is incorrect. Option C, Cruciferae, refers to the family Brassicaceae, also known as the mustards and cabbages family. So mustards and cabbages are present in Cruciferae, and most of these also have C3 type of plants. So therefore, the right answer is option D, Graminae, in which sugarcane, maize, etc. are present. Now note that not all monocot plants are C4. It's just that most of the C4 plants are also monocots, so they are advanced. And most of these advanced plants are found in one family, which is given as Graminae. And that's why we were able to prove that Graminae is the correct answer. Option D is the right option. Now, let's look at this question. In the electron transport chain during terminal oxidation, the cytochrome which donates electrons to O2 is cytochrome B, cytochrome C, cytochrome A3, cytochrome F. So the cytochrome which donates electrons to O2 after engaging in a network of reactions will also be given as the last cytochrome in ETS. And the ETS by itself is a transport chain by which electrons are transported throughout the cell after photosynthesis. So let's embed the picture for electron transport system. Now, as you can see, cytochrome A3 is the final cytochrome present in the ETS, and it donates electrons to O2 in order to form water. So therefore, option C, cytochrome A3, is the right answer. Now, what do the other cytochromes do? Cytochrome B, it is used to convert energy to ATP. So therefore, it's incorrect. Cytochrome C is used to carry electrons from one complex of proteins to another. And these proteins are present in the membrane. So one complex of membrane proteins to another. Cytochrome C carries the electrons. And in cytochrome F, what it does is that it acts as the electron transport connector between PS2 and PS1. So in most plants, both PS2 and PS1 work in tandem in order to create non-cyclic phosphorylation. And cytochrome F is the connector which transports electrons between PS2 and PS1 when they undergo non-cyclic phosphorylation. If this connector is not present, then PS1 acts alone in order to form cyclic phosphorylation. So therefore, we can see that option C, cytochrome A3, is the right option. First proof, we embedded a picture and we found out that cytochrome A3 is at the final position of the ETS. And in the question, it's also said during terminal oxidation. So therefore, terminal refers to the last. So the last part of oxidation would contain cytochrome A3, donating electrons to oxygen. And the other way by which we found out is by finding out what the other cytochrome's functions truly is. And therefore, we eliminated cytochrome A3 as the right answer. So both ways can be done about this. One thing to note is that cytochrome A3 is the final cytochrome which is present in the ETS, and it is present in terminal oxidation as given in the question. So therefore, terminal oxidation would give you the clue that it's the final cytochrome, and the final cytochrome in the ETS is cytochrome A3. So its function is to donate electrons towards oxygen. Option C is the right option. Now, let's look at this question. Which one does not differ between a C3 and a C4 plant? So we have 5 points. We need to find out which of these differ between C3 and C4 and which of these do not. So let's draw 2 columns, C3, C4. And let's put in a column for serial number just in case. Serial number 1 refers to the first point, the initial CO2 acceptor. In C3 plants, the initial CO2 acceptor is RUBP, ribulose bisphosphate. And when the CO2 is accepted, it then undergoes fixation using the enzyme rubisco. So this is found in a C3 cycle and in C3 plants. However, in C4 plants, while this process also occurs, the initial CO2 acceptor is actually PEP, or phosphoenol pyruvate, as given in the first question. And the enzyme which is used to fix the carbon dioxide which is accepted by this CO2 acceptor is pepcase, or phosphoenol pyruvate carboxylase. So as you can see, this point, the initial CO2 acceptor is different in C3 and C4 plants. Now, statement number 2, comparison number 2, extent of photorespiration. Now we know that since rubisco is present in C3 plants, they undergo photorespiration very often. So therefore, we can write that there is complete photorespiration present in C3 plants. However, in C4 plants, due to the special leaf anatomy known as Kranz anatomy, pepcase is used to initially fix the carbon dioxide, and then it's converted into an acid, and that acid is put into the bundle sheath cells, where in the absence of oxygen, the actual C3 cycle takes place. So therefore, photorespiration is 0, none, nada. So there is no photorespiration present in C4 plants. So extent of photorespiration also different. What about number 3? Enzyme catalyzing reaction that fixes CO2. So again, the enzyme catalyzing reaction that fixes CO2. So we just need to look at which of the enzymes are present here. So in terms of enzyme, C3 has rubisco from the start, while C4 has pepcase along with rubisco at a later stage. So therefore, statement point number 3 is also different in both C3 and C4 plants. Number 4, presence of Calvin cycle. Yes, Calvin cycle is the whole cycle which is present in C3 plants. And in C4 plants, after the initial fixation through the mesophyll cells, the carbon dioxide which is present in the bundle sheet cells also undergoes the Calvin cycle or C3 cycle. So C4 is an amalgamation of both C3 and the extra fixation of carbon dioxide in the mesophyll cells. So this point is same. Both have the presence of Calvin cycle. And number 5, the anatomy here is normal, while in C4 it has a special name known as the Kranz anatomy, containing both mesophyll cells and bundle sheet cells in separate ways so as to do separate functions. So the 5th point is also different. As you can see now, only the 4th point is same in both C3 and C4 plants. And therefore, only statement number 4 is the right answer. And which of these options convey that right answer? Option B. Option A says that 1 and 5 do not differ, but they do, so it's incorrect. Option C says that 2 and 3 are not differing, but they do differ, so that is incorrect. Option D says that 2 does not differ, but the extent of photorespiration varies, and it changes very contrastly between C3 and C4. C3 has the extent of photorespiration completely, while C4 does not have any trace of photorespiration. So therefore, option B, which is statement number 4, it does not differ between C3 and C4, is the right option. And now we've reached this question. The total requirement of ATP and NADPH for each molecule of CO2, which is fixed and reduced in photosynthesis in the Calvin cycle, that is C3, is 2 ATP, 2 NADPH, 2 ATP and 3 NADPH, 3 ATP and 2 NADPH, 4 ATP and 3 NADPH. So it is noted that in the whole Calvin cycle, one carbon dioxide is undergoing the same Calvin cycle 6 times, and the final number of ATP and NADPH is 12 ATP and 18 NADPH. So this is for 6 CO2. So if we had it for 1 CO2, then it will be 12 by 6 ATP and 18 by 6 NADPH. So that means 2 ATP and 3 NADPH will be the total requirement of ATP and NADPH for one molecule of CO2. We know the total amount of ATP and NADPH consumed. We know the total amount of CO2 consumed in one cycle. So one CO2 is fixed, 6 is reduced, and it undergoes this cycle 6 times in order to get a glucose molecule. So therefore, we used this method of called the unitary method, where we divided both 12 ATP and 18 NADPH by 6 in order to get that 2 ATP and 3 NADPH are actually the requirements for each molecule of CO2 fixed and reduced. So that is given in option B, so it is the right option. In option A, the number of NADPH is wrong. In option C, both numbers are wrong. In option D, both the amount of ATP and NADPH are wrong. So therefore, the right answer is option B, 2 ATP and 3 NADPH. This is the requirement for one molecule of fixed and reduced CO2 in photosynthesis. Now we've reached our final question. Consider the following statements regarding photosynthesis. Let's read out the statements. Statement number A, ATP formation during photosynthesis is termed as photophosphorylation. And B, Cran's anatomy pertains to leaf. C, reduction of NADP plus to NADPH occurs during Calvin cycle. D, in a chlorophyll molecule, magnesium is present in phytol tail. This is the end of statement number D. Of the above statements, we need to find out which of these options is correct. Option A says A and B are correct. Option B says C and D are correct. Option C says A and C are correct. Option D says A and D are correct. So in this question, we need to find out which of these statements is actually true. Statement number A, ATP formation during photosynthesis is termed as photophosphorylation. So this statement is absolutely correct because ATP formation happens both in PS2 and PS1. So combination of PS2 and PS1 was given as non-cyclic, while the involvement of just PS1 will give you cyclic photophosphorylation. And it is for this photophosphorylation that electrons are taken from water and oxygen is released. So therefore, this statement is correct. Statement number B, Cran's anatomy pertains to leaf. Now, this statement is also correct. The term Cran's anatomy refers to the special arrangement of cells in C4 plants. In C4 plants, the bundle sheet cells are closer to the vascular tissue, so they get the nutrients and water, while the mesophyll cells, they receive the carbon dioxide and then they fix it in the absence of rubisco so as to avoid any amount of oxygen to go into the bundle sheet cells so that they can have photosynthesis without photorespiration. So statement number B is also correct. What about statement number C? Reduction of NADP plus to NADPH occurs during Calvin cycle. In reality, the reduction of NADP plus to NADPH happens during, let's go back. It happens during the phase of photosynthesis known as light reaction. So reduction of NADP plus to NADPH occurs in light reaction, and Calvin cycle is a part of the dark reaction. So therefore, statement number C is incorrect. Statement number D, in a chlorophyll molecule, magnesium is present in phytol tail. Now, chlorophyll is divided into two parts: porphyrin, which makes up its head, and phytol, which makes up its tail. Now, porphyrin consists of a number of carbon atoms present in a ring, and it has magnesium at the center of the ring. So therefore, the magnesium is actually present in the center of the porphyrin head instead of the phytol tail, as it's mentioned in the option. So statement number D is also incorrect. So now, after reviewing them analytically, we know that statements number A and B are correct. So the option which says that A and B are correct is option A, so that is the right option. Now, option B says that both the incorrect options are correct, so that is wrong. In option C, it says that C is correct, whereas in fact C is wrong. And in option D, it says that D is correct, statement D, which is wrong. So the option is also incorrect. So therefore, after finding out the verification of all the four statements and proving that they are true or false, we found out the right option. And that concludes this episode of NEET preparation. Now, we have done many episodes in NEET biology. So there are a lot of episodes and chapters in biology in this channel called Brain Blitz Audio. So if you want to access that, then please do subscribe to our channel. I've also provided a link in the description down below which will guide you to the playlist for NEET Biology, so you can take our other videos and view them at your leisure. And you can also hit the notifications button if you want to receive up-to-date information about our latest and innovative content, such as mind maps, NEET preparation, and learner words, memory hacks, English pronunciation, and other useful content. So until the next webisode, take care, stay safe, bye-bye for now.